All right, so we're back in the lab or Airbnb, and we're going to show you how to process the water and plankton samples that you collected. So in your sampling kits, you will receive printed out instructions that will sort of guide you through the process, as well as a lab sheet that will also sort of help you go through the process step by step. Two sides. So this is our lab setup for processing water samples. We have a few filter manifolds for 25 millimeter filters, GFFs. And here the water goes straight with the waste, so we're not retaining the water that is filtered through here. We're just keeping the filters. And those filters are used for PON, POC, stable isotopes, and for chlorophyll, correct? Anything else, Drew? No. Okay, and then we have a set of 47 millimeter filter holders that go into these catch flasks. And here we're actually going to keep the filtrate as well as the filters. So the filters you will get in the 47 millimeter filters, you'll get in these glassine envelopes and they have a number and a weight, empty weight for the filter, which we will eventually record. These go on here, and these are going to be used for fatty acid analysis of ceston. And the filtrate that goes through these filters is going to be used for dissolved nutrients, nitrate, nitrite, and for some organic carbon watercolor and EAMS, which stands for emission excitation matrix spectrometry or something along those lines. Yeah, so we're going to take you step by step through all of these things, and we're also collecting water for total nitrogen and total phosphorus, which is not filtered through anything. One note is that all the water that we're going to be collecting is going to be pre-screened through a 200 micron mesh, which I think you will get as part of your sampling kit. Drew is nodding to confirm. So you will get this, and we use this to pre-screen all the water that we then use both for TNTP and for the filtered fractions. So yeah, we'll show you how we go about this process step by step, starting with total nitrogen and total phosphorus and moving on to the filtration. All right, so Drew is going to show you how to do total nitrogen and total phosphorus samples. She has lost her voice, so I'm going to narrate this part. So mix your water sample and pre-screen it into an intermediate container. All your plasticware and glassware should be clean. Okay, and these are 250 milliliter bottles, I think, that we use for TNTP. Again, this water is not filtered, it's just pre-screened. Fill it approximately 90% full, sort of to what people call the shoulder of the bottle, to allow for expansion during freezing. So you're going to collect duplicates of these for each of the 3 depths that you sampled for a total of 6 TNTP bottles. Okay, so we're just going to do it again for the other 2 depths, and then freeze those TNTP samples. So we finished doing the TNTP, and we're going to check that we have done it on the field sheet. So TNTP, depth 1 is the top, depth 2 is the mid, depth 3 is the bottom, and we did both replicates. So we're gonna check these boxes, and if there's anything to note here, we would do so, but there's nothing to note, so we go ahead. Okay, so we finished TNTP, and now Drew is going to set up for chlorophyll A on 2 of the towers, and then POC, PON for CN stable isotope and total organic carbon organic nitrogen analysis. So in your kits, you will receive a bunch of pre-combusted 25 millimeter GFFs, which you will use for both chlorophyll A analysis and for POC, PON analysis. So Drew is going to run those at the same time. And for chlorophyll A, we usually use how many mils?
150.
150. In rare instances, in really productive lakes, you might have trouble getting 150 through, but I don't think we've ever run into this issue, even in our very eutrophic lakes. So I think you can aim for 150 for chlorophylls. For PN, POC, our goal is to get as much water as we can through these filters, which ends up being, what do you think, Drew?
Should about 300.
About 300 in like oligotrophic, mesotrophic lakes. In really productive lakes, you might clog the filter after about 150, 200. So if you're working in a really productive lake, maybe start with 100 and then keep adding water as you go. Important to note that for both chlorophyll and POC, PON, you will need to record the total volume that you filtered. So those are actually, so POC, PON is going to be over here. And I think the chlorophyll is on the back. Chlorophyll is over here. I can't remember the volume. So in a setup like ours, where you have access to both 25 millimeter and 47 millimeter towers, we don't mind sort of not using this water and letting it just go to waste, because we're going to generate a lot of water through our 47 millimeter filters. If for some reason you are using syringe filtrations or don't have 47 millimeter filters, you will probably want to save the water that you're running through the 25 millimeters. Okay. So again, Drew is pre-screening her chlorophyll A sample and measuring 150 milliliters into each of the two replicates for the first water sample, which is our top water sample immediately under the ice. And chlorophyll A samples will go into these 15 milliliter Falcon tubes. And I hope you will receive all your plasticware pre-labeled, so it will be pretty straightforward, unless we run out of time. But again, I hope you'll get them pre-labeled, so it will be pretty easy. So you see, the lake name, which will generate for you based on the information you enter, the depth, which is top, and then replicate A for chlorophyll A. Notice that there is a spot here on the Falcon tube to write down the volume that you filtered. So you'll write down the volume that you filtered both on the tube and in your field sheet. And Drew is dispensing 150 mils into each. So I'm just going to write that down in our chlorophyll A spot here. Okay, and you're doing 250?
I'm starting at 200.
Okay, so Drew is starting with 200 for the POC-PON, and if the 200 passes through easily, we're probably going to bump it up to 250 or even 300, depending on how fast it's going. And the POC-PON samples will go into little Petri plates, I believe, correct?
Yes.
So you will get little Petri plates that look like this, and will also be pre-labeled for you, and you'll also record the volume on the label. We'll show that to you in a second. So while Drew is filtering the chlorophyll A and POC, we're going to set up for the sulfate fatty acids, which we're going to keep on the filters, and the filtrate, which we'll use for a few different fractions. So again, like I mentioned, you'll get numbered glassine envelopes that will have an envelope number and a weight of the empty envelope, which you will record in your sheet. So this will go over here, and I'm going to record 3 pieces of information here. I'm going to record the total volume that I'm actually going to run through it, which I'll mention in a second, and then the envelope numbers. So for top replicate A, I'm using filter 207, and it weighs 1231 grams, and for replicate D, I'm using filter 206, which weighs 1206 grams. Perfect. Thanks, Drew. So we are going to load them, and once you've written this information, you no longer need that to start that. So I'm going to measure some water, and we're going to run it through Typically, because these filters are much larger, we can run somewhere between 600 milliliters to maybe 1.5 liters through these. Our goal again is to load the filter as much as we can to get a good signal for fatty acid samples. So this is a sort of mesoeutrophic lake, at least in the winter, so I'm going to probably start with running about well, maybe 700 mils, and then just keep adding once the 700 mils go through, because it really sucks to overload your filter funnel and then wait for half an hour as the last 50 milliliters go through. So at the end we add little by little to top it up. Okay, so Drew filtered 150 milliliters through each of the chlorophyll filters. You can see they're getting a little bit of color development, and now she's going to put them into the 15 mil Falcon tubes. Folding it in half and putting it in. Same thing for the duplicate, and she will write down 150 on them when she's done, and then those will go in the freezer for storage. Okay, I am filtering through the 47 millimeter filter, so I measured 700 mils of pre-screened water into my graduate cylinders, and I ran 200 milliliters through these filters to rinse the flask. So what I'm going to do is I'm going to turn these off now, hand the camera over to Drew. Take these off. And use the first 200 milliliters to do a little rinse for the flask. And now I can dispense the other 500 mils that I measured. So I would have filtered, once this is through, I would have filtered a total of 700 milliliters. And once that's through, we'll see if we can add another 200-300 for a liter. So that will depend on how fast these two replicates go. Again, normally we'd be filtering a couple of depths in parallel, but we don't want to confuse ourselves with overstocking, so we're just doing it one at a time for the video. Okay, so we were able to push 300 milliliters through these 25 millimeter GFFs for POCPN. Just want to show you the color. So what we're basically looking for is color development, good color development on the filters, and that should be enough material. Okay. So these will now go into these little feature plates, and Drew will write the volume that she got through over here, and also on this sheet here. So this is our top depth. Replicate A and replicate B both got 300 milliliters. And this is the 47 millimeter filters. I added another 300, so I'm hoping I can pass a liter in total for each of these 47 millimeter GFFs. Okay, so we now pushed 1,000 milliliters of water through these 47 millimeter GFFs. Took about 10-15 minutes. So you can see there is some color development on them. Hopefully that will be enough material for fatty acid analysis. So what I'm going to do now is I'm going to put the filter into these 4.5 milliliter cryo vial. Notice I wrote down the total volume that I filtered. So what I'm going to do is gently roll this into a nice little sort of burrito, crepe, some other food product that's rolled, helping myself with the little vial, and stuff it in the vial. One replicate, and then here is the other replicate. Okay, and these should be frozen in the coldest temperature that you can manage wherever you are. We have a liquid nitrogen dry shipper, so I'm going to throw them in there. Okay, now we have the filtrate, which we're going to use in a few different ways. Okay, so first replicate, oh, and by the way, I also wrote down the total volume I filtered for fatty acids over here, 1,000 mils. So we are going to fill this large 500 milliliter bottles for dissolved phosphorus and ammonia, and we're gonna try to get it about 80–90% full, something like this is enough. And these bottles, when you receive them, will be pre-labeled and also cleaned, so you don't have to clean them. Okay, another sample for nitrate nitrite, 80–90% full. And then you will get 3 of these 40 milliliter amber vials, like I mentioned, for EAMS, for color, and for dissolved organic carbon. So these you will fill, again, about 80–90% full. Right, we'll cap them with these subtate caps. And these 2 samples, dissolved nutrients, will go into the freezer. EAMS will go directly into the fridge. And the color and DOC samples you will preserve by adding 40 microliters of 6 normal HCl to these, and then they will go again in the fridge. So all the glass vials go in the fridge. These 2 get preserved with 6 normal HCl, 40 microliters. This one is not preserved with anything, it's just filtered and placed in the fridge. These go in the freezer, and I'm going to do the same thing with my duplicate. And then we're going to do the same thing with the other 2 depths. Again, processing everything in duplicate.
Hi everyone. So we just came back from the field, and remember the samples we took for zooplankton, the 80 micromesh, and that for the 35 micromesh. Now we are going to fix it with logos. Now for the 80, we need 10 mils of logos, and for the 35 micromesh, we need 5 mils of logos. This is 10 mils. Make sure you don't allow the tip of your pipette to touch the water. You do the same to the duplicate. Okay. And you cover. Do same for the 35. And this for this one, you just put 5 mils. Okay. Okay, and that's it. For the other water chemistry, you need whole water samples to fill unfiltered whole water samples to fill the bottles for the flagellates, the phytoplanktons, and the ciliates. Both phytoplanktons and ciliates need glucose, and we need 6.5 mils for the phytoplanktons, and you need 2.5 mils each for the ciliate. But for flagellate, you need 6 mil of 37% buffered formaldehyde to fix this sample. So I think with that, you make sure you do all this operation in a well-ventilated environment, and make sure you paraffin this to avoid what spillage. Thank you.
Okay, so it's been about an hour and a half. We finished processing almost all of the water samples, so from the 3 depths in duplicate. There's a couple of things left, and one of them is processing the zooplankton sample that we collected for fatty acid analysis and stable isotope composition. So we've had it sitting in the fridge upright for the last few hours. I noticed that there is some stuff that has sort of sedimented at the bottom, which is like detritus and other gunk that we don't want in our samples, and most of the sort of overlying water is composed of live zooplankton. So what we're going to do is now very carefully screen this sample, trying not to resuspend the material at the bottom, through that 200 micron mesh sieve that you've gotten. So at this point you don't need it for water anymore, and we're going to use it to concentrate the zooplankton sample. So again, I'm sort of gently pouring it, trying to not resuspend the material at the bottom. Again, this is a pretty thick sample, so I don't have to screen a lot of it, and I can be pretty conservative with how much material I'm passing through, avoiding resuspension. And again, notice how thick this sample is. It's just a ton of big Daphnia and big calanoid copepods, a lot of cyclopoids as well. Kind of a neat sample. Again, sort of gently decanting it. It's probably quite good. Yeah, check it out. Let's look at this zooplankton soup. Tons of material. So what I'm going to do now is I'm going to give it, come back here, pipette. I'm going to give it a little blast with DI water to sort of push any small stuff that's sub 200 micron through, and also to concentrate the zooplankton in the corner here. Right, and then I'm going to get 4.5 mil cryovial for the zooplankton fatty acids, and using one of those pipettes that will provide you with sort of almost really, look at this zooplankton goop, kind of like scoop it in there, really. It's thick enough that I almost can't put it into the pipette. I'm just like pushing it in. And, you know, this is like way more material than we need. Like, really what you're sort of aiming for ideally is like this amount of biomass, and again we have way more than that here. So there we go. And I have a duplicate bottle in the fridge, but I think I'm just gonna use this just because there's so much material for my duplicate sample as well here. Way more than we need. Here's the lid, and just like with the Seston fatty acid sample, we're going to freeze it at the coldest temperature that's available to you. So here, like I mentioned, we have a liquid nitrogen dewar. Sometimes we're working in the lab and we have access to a -80, so we'll put it directly into -80. If you don't have a -80 or a liquid nitrogen dewar, you can freeze it on dry ice, for example. So the coldest that you can basically manage before storing it at -80 when you get to the lab. And next we're going to show you how to process the DNA samples for microbial community composition.
Hi, so now we'll be doing microbial DNA composition, and I will just give you a brief list of things we need to have before we can do that. So first off, you need ethanol, which you need to, sterilize your top. You need your pre-label vacuum tube. You need your sterile clean Sterovex. You need your parasitic pump and the tube. So if you don't have a parasitic pump, use a syringe, which I will show later and how to use it. You need clean molecule water, and you need your sample water. Okay. So before you start the whole procedure, after wiping down the surface, you make sure you clean your tube by running about 100 to 200 mils of this milky water in through the pipe to make sure it's properly clean. So you run clean milky water through the pipe about 100 to 200 mil to make sure it's properly clean before you insert into your sample bottle. So now you attach your Sterovex filter to one end of the tube and the other end into your sample bottle, and you filter till you keep passing water through till your Sterovex filter gets clogged, and you do that very slowly so that you can get much water through it. And then you make sure as the water comes out through the Sterovex, it goes into a container which is here, so that you can easily measure the total amount of water you filtered through using a cylinder. Now we can see that we have one tube into the sample bottle, another one with the Sterovex into a bottle, so we can get the filtrate out. So we can measure the total amount of water you filter through the Sterovex. So we continue doing this until the Sterovex is clogged. So we are, like I said, we are aiming for a filter of this color. So this has been clogged and water is just seeping through very slowly. So we cover the first end of the paraffin of the filter with paraffin, and then you cover the second end also with paraffin to prevent any contamination. Then once this is done, you make sure it's tightly covered, and you store in your pre-labeled 60 ml 50 ml vacuum tube this way, and you lock it, and then you freeze this. If you don't have if you don't have a -80 or a -20, you can also freeze this in your with your liquid nitrogen, just like this. Thank you. Yeah, good. So you also take the filtrate and measure the total amount you pass through the Sterovex. You can do that, so you can measure that using a cylinder. Alright, so we can see we did close to 1,000 ml, and you just record that on the Sterovex. If you don't have a parasitic pump, what you just need is a new clean syringe, 60 ml, and your Sterovex filter. You attach this, you make sure the syringe you get is a Limnology syringe that connects to the Sterovex. You connect add your sample water and what apply pressure to what push it out. You do that as much as possible until the styrovet gets clogged, and make sure you record how many amounts you pass through the styrovex. Thank you.